Hello, everyone, and welcome to today's Keynote webinar titled Quantifying Biological Parameters with Online Flow Cytometry, Boosting Bioprocesses by Providing Single-Cell Data in 24/7 Resolution. This webinar is part of the 7th Bioprocessing Virtual Event Series. I'm Matt Thorne of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots. Before we begin, I'd like to remind everyone that this event is interactive. We do encourage you to first participate by communicating with other attendees using our live chat feature during the presentation. You can find the live chat located in the bottom left of your screen. You can also participate by asking as many questions as you would like during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We'll answer as many questions as we have time for at the end of the presentation. If you have any trouble seeing or hearing the presentation, click on the Help Desk button from the lobby within the event. Finally, as a reminder, this presentation is educational and offers free continuing education credits. Click on the continuing education credits link located within the abstract on the left of your screen and follow the process to obtain your credits. Now, let's go ahead and get started. I now present today's speaker, Konstanze Schiessl, Chief Scientific Officer at onCyt Microbiology. For a complete biography on our speaker, please find them on the left of your screen. Konstanze, you can now begin your presentation.
Okay, thanks a lot. Thanks for everyone for joining and listening to my presentation. I don't know where you're all located, but I appreciate you taking the time. I myself am sitting in Zurich in Switzerland at the moment, and I will tell you about something that I work on every day, and that is online flow cytometry. In the end, this talk will be the introduction of a method and then some use cases of using this method in different types of bioprocesses. And the method that we are going to talk about today is called flow cytometry. I will explain it later in a bit more detail how it works exactly, but you can already see an overview drawing up here. So you see there are things that's called laser detection. You see a lot of light going around. So indeed, we are talking about an optical detection method. It is called flow cytometry because it uses a water stream where cells are separated within. And cytometry, it comes from measuring the cell. So we are indeed talking about a technology that's focused on biological particles and on measuring them using a laser and an optical detection. Flow cytometry has been a standard method in medicine for over 50 years, and you will find it in many labs everywhere. But it's becoming more and more also a method that's used in industrial processes by streamlining it, by making it more user-friendly, by shrinking the size of the machine. You can really also bring it outside of strict academic or diagnostic applications. And this is what I'll be focusing on today. So there's just some terms or key concepts that I will mention in this talk, and I wanted to already introduce your brain to them. So here I wrote, I will talk about use cases in bioprocesses. Here in the middle, on the left, you see pictures of different organisms that are all measured with flow cytometry. So on the top, you see CHO cells, Chinese hamster ovary cells. So these cells are used to produce many advanced drugs like monoclonal antibodies. And the flow cytometer can measure them. So we will talk a little bit about that today. Below that, actually, you see CAR T cells that are attacking a cancer cell. So also in CAR T production, so very advanced bioprocess, you could say, the flow cytometer plays a key role. And towards the middle, you see microorganisms, so the different classes we have here. So we have microbes and like bacteria themselves, then we have yeast and microalgae. And all these three I will tell you examples about and how we used online flow cytometry to measure these. So you see that the flow cytometer can be used quite diverse across biological particles. And then one key term that I think is important for, well, why do we care about this method at all, and why is it important for bioprocess, is the concept of process analytical technology, or PAT technology. Before I go into all of these many topics, I really briefly want to introduce my company, where I come from. So yeah, we are called onCyt, and what we do is we indeed develop process analytical technology using online flow cytometry, and we are focused on monitoring biological parameters. I want to mention that a lot of the data I showed today has also been funded by the EU in the Promicon project. And here in the middle, you see a data set. So this has been collected with an online flow cytometer. You see on the y-axis, it says cell concentration. So here, actually, we have a data set from a cooling tower. Cooling tower has water, it has heat, it has some nutrients. So bacteria love to grow in these conditions. And the people responsible for the process need to make sure that bacteria don't grow too high, can lead to Legionella problems or other types of problems. And we put our online flow cytometry device in there, and it gives you really nice data on how cell concentration changes over time. So you see an increase and then a sharp drop. And the sharp drop is because at that moment, that the manufacturer put a biocide into the process, and you see it kills the cells quite fast. But you also see that the cells immediately start growing again. So this is the type of data you can get with an online flow cytometer. So it really is it's a glance at what we will look at later. And this is what our company does. So we work with different sectors and develop these types of machines. So before I go into the data sets, I did want to spend a couple of minutes on the PAT concept itself. So this is going to be the first part. Then I will talk about why flow cytometry is a great tool for that. I mean, there are also other tools, and then I will mention them. So you will learn a bit about what sensors are available. And then the last part, and by far the largest one, will be showing data sets or publications or examples of the use of online flow cytometry for process monitoring and optimization. So this is a bit theoretical and a little bit dry, this slide, but I think it has some important motivational information to understand why we care about tools like online flow cytometry or other sensors to monitor bioprocesses. So PAT, this thing called process analytical technology, it actually has been defined by the FDA. So it's a really important concept for industry, and it's a mechanism to design, to analyze, and to control manufacturing processes by measuring something called the critical process parameter. And these affect critical quality attributes. I will get into that a little bit more later, but these are kind of key terms that you will come across in the manufacturing space. And these things are there so it's really about understanding the process and then monitoring the process in a really fast manner. So preferably inline or online. So really getting the data the moment, in the moment, not like 1 hour later after you did an experiment in the lab on it. And the motivation is to improve the production process, also the safety of the process, especially in pharmaceutical manufacturing. That's really important. So PAT is a real-time testing, and it's based on the complete understanding of how the process actually affects the final product. So let's say in the old times, maybe you just measured the final product, and then either it was fine or was not fine. But now it's more about really measuring all along the process and knowing in real time if something is going to be wrong in the product. And then, of course, you can also react a lot faster. So let's quickly talk about the CPPs. So these are these things that critically influence your process. So it's a process variable that has an impact on really important quality attributes, and these things need to be monitored. And nowadays they are also monitored really in real time. So typical critical process parameters are temperature, pH, dissolved oxygen, nutrients, and metabolites. So these are physical and chemical parameters, and I'm sure you all know about, pH sensors. So these things are really standard in manufacturing processes. And then we have this term called critical quality attribute. So this is more about the product already, or closer to the final product. So it's defined very broadly as physical, chemical, biological, or microbiological property that should be within an appropriate limit. And this is all defined depending on the product. The thing is that these CQAs, they are a lot harder to measure in the process than the CPP. So pH and temperature, it's fairly simple and fairly cheap to monitor online, but the CQAs, it's a lot harder. Examples of CQAs can be, variations of the product, like a cell phenotype. So in CAR T, for example, this is really important. It can be details of the product, like the glycosylation of a monoclonal antibody, but it can also be the bioburden, so the microbial, let's say, contamination of a product. Okay, so it goes on that there's not just the CQAs, there's also the key performance indicators. These are also important parts of understanding the process. And they should indicate that each of the production steps proceeded as expected, so that in the end you will meet your critical quality attributes and they will be within range. And so these ones are also harder to measure. So this can be product quality, product titer, like how high is the concentration, but also things like total and viable cell density, for example, of CHO cells in monoclonal antibody production. And other things highlighted here in green are things that a flow cytometer can measure. And so you see we are not here in the top range of fairly simple sensors for CPPs. We are more in the range of kind of complicated parameters that are already closer to the outcome of the process. But these things are typically really hard to measure automated and online. So nowadays, mostly they are measured with like manual methods. So someone taking a sample, bringing it to a lab, processing it there. So it's really challenging nowadays to measure these important parameters in an online way. I wanted to add one quick slide because I don't know if everyone understands when I talk about online. Many people think of online like the internet, but it's actually also here in this PAT concept, it's also about a way of measuring. So there are different types of measurements around a process. So for example, here we have a bioreactor, and you see in the schematic what these different types of measurements are. So on the right, you see an offline measurement. So that really means someone goes and takes a sample, brings it to a lab, and then it's analyzed there, for example, in a HPLC. So this is not ideal in terms of PAT, where you really want, around the clock rapid measurements. But for some methods, that's just what is available. Then you get one step closer to the process, which is this @line thing here. So this is often, a method that you can, you still take the sample out of the reactor, but you can process it kind of close to the reactor. Maybe it's a fast, like a rapid test that you can do there on a bench, and then you have it fast and it's close to the infrastructure. And then there's the online thing that you see on the top left. I'm sorry, I can't point, but I hope you can follow me. So here you take a sample out of the reactor, you bring it to the measurement device, and then it's typically not always, but typically circulated back to the reactor. So here you're really close in the process, and often this is also automated, so it doesn't require anyone going there and taking a sample. And then the one that's really the closest to the process, let's say it's the sensor is directly in the bioreactor, directly suspended in the sample, like a pH sensor would be an example for that. And so, yeah, like it says above, for PAT, it's preferably inline or online. Okay, good. Yeah, so this is the last slide on the PAT concept. And I just wanted to give an overview of parameters that are typically measured in bioprocesses. What type would it be? Is it a CPP or a KPI? And how are these parameters measured to give you an overview of the state of the art nowadays? So like I already said, pH, dissolved oxygen, glucose, and lactate. So these are typical CPPs, and the measurements methods, they vary a bit. So I think the easiest ones are pH and dissolved oxygen of the examples here. And you can see like pictures of these on the right. So these are typically super easily integratable into a bioreactor, and they really measure inline. It becomes a little bit more complicated with the metabolites, nutrients, so glucose and lactate. So there are several methods, several spectroscopic methods available, like Raman is very famous nowadays. And that can indeed also measure inline and online, but it always needs a calibration. So it requires a bit of tailoring to each of the bioprocesses that it's used with. And there are also a lot of offline methods for these metabolites, like HPLC, for example, but there you really need to go to the lab. And then we move down, and now we move to a KPI. I think in some processes it's also CQA. For example, if you grow probiotic bacteria, this is already a product, so it's really important to know total and viable cell density. And there are many methods out there. I'm sure you're familiar with some of them. So there are offline methods. So I think these are these are widespread depending on the process. These are cell counters. There are different manufacturers that offer trypan blue staining. And then there's also different sensors for total or viable cell density using different types of optics. And these can indeed measure inline. So I think these are becoming more and more widespread used. But I think in many cases, the gold standard is actually still offline measurements. Okay, and then we move down here. I put an example that's cell cycle. So this is, a parameter you don't need for any bioprocess, but it can be interesting, for example, for monoclonal antibody production, because it can be related to the production itself. So it can be a KPI. And this, for example, can, as far as I know, only be measured with flow cytometry. And nowadays, this is done offline. So you take a sample and bring it to the lab and analyze it there. Then another one, since I love to talk about microbiology, so bioburden, that's important. What's the microbial load? And nowadays, these things are also done offline, like plating or qPCR, like cultivation dependent or replication dependent methods for that. And then the last example is really, really advanced. So this is like a very detailed cell phenotype. And I think this is mostly relevant for cell therapy, where the cell is the product itself. And also here, to my knowledge, there's no other method than flow cytometry to be able to quantify all of these details. And nowadays, this is also always done offline. And I marked the offline here in red because it's from the view of PAT, right? You really want to have a full process understanding and process control. And for that, you should always aim for inline or online methods. Okay, so in summary, let's think of it in a schematic, right? You see here data over time, so it can be just some type of bioprocess. Think, for example, this data could be cell concentration. And if you have an offline method, you take maybe 4 measurements over time, but the bioprocess is not stable. There's always some variations and dynamics going on, and you might miss important dynamics of your process if you don't have an online or inline system that really measures around the clock. And yeah, for the KPIs and CQAs, most methods are still offline. And we believe that flow cytometry is one method that can measure KPIs and CQAs online. And I want to explain a bit why, well, what is flow cytometry and why is it nice for these applications. So on the left, you see a schematic of how the measurement method works. So you load a sample, it's suspended in liquid. So you see here the blue and the red cells. And then a thing happens within the machine, which is called hydrodynamic focusing. So I don't fully understand the physics of it, but in the end, you have your sample stream and it's guided into a core of a fluid, which is called the sheath fluid, which is in the end just super clean water. And then the cells interact with this sheath. And then in the end, what happens is that in a very thin channel, really the cells are separated physically. So you have one cell flowing after the other. And then these cells pass by a laser, and the laser can interact with the cells. And for example, often fluorescent staining is used to stain specific molecules of the cell. And then these signals can be collected with different detectors, and many properties of each single cell can be quantified. The benefits of flow cytometry are that it's really fast. So yeah, time to result is typically minutes. If you need to stain and incubate, it takes a little bit longer, but it's really, yeah, we're on the minute scale here. It's very representative compared to, for example, plating. So really every cell is measured, growth independent, and we have high throughput. So thousands of cells are measured per second. Yeah, again, compared to plating, you have automated detection, so it's less error prone. And a really nice thing about it is that you can use different types of staining assays, and I will mention some. And this really gives you a lot of detailed biological information of the cells that not many other methods can give you. Overall, I think it's still fairly simple to use, especially the newer models. Well, maybe some people disagree, but I think, well, you need to know how to set up your measurement, but after that, it's kind of simple to use, I think. It can be automated, which is really nice for the online and the inline. And one thing I do want to mention, and people here might know about that, that flow cytometry can also be combined with sorting of cells. So, once you have your cell characterized, you can also say, I want, for example, all viable cells or all cells that have a certain biomarker. I want these in a separate tube, so they can really sort the cell population into the cells that you want or don't want. And this can be super valuable for, I don't know, selecting clones or sequencing or things like this. And this is often called FACS. So many people might say FACS and flow cytometry is the same thing, but it's actually not. So the flow cytometry itself, it's just the analyzer that I'm showing here, but you can combine it with sorting. And this is not something that we do, so it will also not be in the examples, but it's a really cool feature of flow cytometry. Okay, and then there are many, many machines out there. So there are many offline machines, like really, really many. I just put a couple here. And if you ever worked in a lab, I'm kind of sure you already came across flow cytometers, and they can be huge with hundreds of lasers and detectors and so on. And what we do is that we actually have online flow cytometers, or maybe to be a bit more precise, it's automated at-line. So we take the sample from the bioprocess, we prepare the sample automatically in our devices, we feed it into the measurement unit, we measure it, we analyze the data, and in the end, you get these time curves that I showed before. And this is something that we have developed, so we really bring flow cytometry in this online state. So the way we do it, well, we connect to a bioreactor, so you can connect up to 6 sample lines, and then we sample. We have a patented method for that. Then automatically the sample is diluted if it's needed. For example, for microbes, that's always important because the flow cytometer has a limited linear range. And then you can automatically stain with various assays, as I will show. And then, after the staining is prepared, has been incubated, we feed it into the flow cytometry unit and measure it, and then you get automated data analysis. Then it looks a bit like this. In the end, you see, for example, concentration of different total and viable cells in different bioreactors. Okay, so now I need to change gears a little bit, and I've not practiced the transition, but let's say the last part, I think we're around halfway through. The last part is really about showing you use cases of the online flow cytometer, and I will give you some examples on things that we have worked on, some publications, some customer projects, and we, like most of the examples I will show about microbiology, because this is what we've historically been doing most. And at the end, I will touch on monoclonal antibody production. And yeah, feel free to ask any questions. It's a bit of a mixed bag. Some things will be more detailed. Some things are just a glance, and you can look at the publications if you're more interested. So what you see here, it's a important part of the work for flow cytometry for microbiology. So what we typically use the flow cytometer for in microbiology is to quantify total and intact cell concentration. So how many cells are in your sample and how many of them are approximately alive. So intact is not always the same as viable, but it can be for many applications. And using a flow cytometer for microbes, it's not that straightforward. So mostly it was used for mammalian cells and for diagnostics. So the idea to use this method also on really small microorganisms has been the last 15 years, I would say. And this comes from Frederick Hammes' lab, which is from the Water Research Institute in Switzerland, and he really pioneered this work. So he figured out ways to stain the cells for total and viability. It just takes 15 minutes, and yeah, you get a really high resolution. So here now we show a data plot that you actually get with a flow cytometer. So typically what you plot is you plot, for example, fluorescence. So remember there's the laser that excites the molecules in the cell, and then you collect the fluorescence that is emitted from there. And here we typically plot green over red fluorescence, and you see different clusters. So every dot here is a particle that was measured by the flow cytometer. And the darker it gets, the more concentrated in fluorescence these particles are. So you see these clouds, you can call them. And this is from a tap water. So if you turn on your tap, there are bacteria in there. They're around 10 to the 5 per milliliter, which don't worry about that. It's totally fine, totally normal. This is a typical, let's say, fingerprint. So if you look at the literature here, this is typically what you measure with a flow cytometer, and you can differentiate between larger and smaller bacteria. And this is really, at least in Europe, wherever you go and measure tap water, this is what it will look like. In the US, it might be a bit different if there's chlorine in the water, but in Europe, this is what you typically get. And we use here different staining methods. So I wanted to at least introduce one staining method. So you get one example in a bit more depth. So for the total cell count, what we use is a stain that is called cyber green. It's also used for PCR. So maybe you know about it from other contexts. So it goes into any type of cell and it binds to nucleic acids, as you can see here. And it can be an intact cell or a damaged cell. And then you can excite it with the 488 laser and then collect the signal at 530 nanometers. And then you have a second stain that you can add, which is called propydium iodide. And this one only enters cells that have been damaged. So you can actually excite it with the same laser, but it will, the emission will be in a different channel. So you can differentiate the green from the red one. And the principle of this is shown in this schematic. So you see if it's an intact cell, the PI can actually not go inside. So the intact cells will not accumulate any red fluorescence. They will just have the green from the cyber green binding. If the cell is damaged, however, the PI can get inside and it also binds to nucleic acid. So there's a bit of a competition between the cyber green and the PI. And this permeabilized or damaged cells, it will then be red because of the PI and also a little bit green because of the cyber green. So for all types of staining, there's always some biological mechanism underlying it that you exploit to get a good signal in the flow cytometer. So now how can we translate this staining into getting the total and the variable concentration? So this is shown here. So what we typically do is we first stain only with cyber green. This is what's shown in plot A. So here you have cyber green only, so any cell total or intact or damaged, it doesn't matter. It's stained, it's turning green. So it increases here in the green fluorescence, and we can identify this region in what we call a gate, and we call this the total cell concentration. And then we take the same sample and we stain it with a mix of cyber green and PI. And what now happens is that the damaged cells, they take up the PI and they increase in red fluorescence, which means they shift up on this plot here. Please note that the red is on the y-axis, so they will increase in red. And at the same time, they will also decrease a little bit in green because cyber green and PI, they compete for the binding sites. And this means now you can really nicely see a separation of the damaged from the intact population. What we often notice is that actually the damaged and intact, as you can see here, there are also different clusters in the green even before you add the red. So you can really see often some like a zoom into your population that you might not see with other methods because the stains, they bind to biological molecules and this often reflects real and also relevant heterogeneity in your populations. And so for every staining assay, there's a logic behind it, and then there's a logic in how you gate, so how you separate these different populations. And this, I think, is one of the parts that's a bit hard about flow cytometry. So you need to make sure you have good controls and you understand how to run the sample. But once you've set it up well, to automate this and to repeat this, it's very easy and fast. Okay. So why do you even use this online flow cytometry? It's we have seen that really wherever we went to measure over time using the flow cytometer, we see fluctuations. So microbes, they really fluctuate everywhere. There are by now 24 publications out with our technology and across different microbial areas. So biotechnology, drinking water, microbiology, where we originally come from, wine production, co-cultures of microalgae and bacteria, but also marine ecology. So someone took our device on a cruise in the Antarctic and measured how the populations changed as the ship moved along. Flow cytometry is also interesting for optogenetics or probiotics. And I will explain some of these examples. And I will do this in a very, really broad scale. There's always a publication listed, so if you want to learn more about it, then you can just check out the publication. And the logic of the slides will always be that you have these microorganisms. Example will vary, and they will fluctuate in a process due to a driver, let's say that we might have identified. Okay, let's get started. So I think one of the earliest examples that we looked at was in rivers. So we really measured from a river and followed it over several weeks. And you see how the concentration really changes. And you see these two peaks here. And this was after a rain event. So when it rains, the system is perturbed, and suddenly you have these peaks of, well, can be up to tenfold in microbial concentration. Here we have a biofilter. So this is in a more industrial process in water production. And here you see these patterns that are characteristic of this process here, and it's linked to how the process, how these biofilters are operated. Here we have another example. It's the tap water I already mentioned. So you can simply hook up our system to tap water and see what happens. And you see here that, yeah, indeed, it can be up to 10 to the 5. And that peak that you see here, that jump, that was Monday morning. And it was because on, the weekend people don't use it a lot. So it stagnates, and then you have this increase here. Here's another type of water system where we have suddenly very high peaks in an otherwise quite stable environment. This is an example from water reuse, so gray water reuse, which means like reusing water from washing machines or sinks. So it's and yeah, there's also logics to this process. Here we have again the cooling water example I showed at the very beginning, where you see these sharp drops and then increases, and these drops are due to the addition of biocide. Okay, here we have actually two different populations, and these are consortia of microalgae and bacteria. And they were grown in a reactor, and then, at some point, one population can take over the other one. And you will see that in the flow cytometer. Okay, now let's go a little bit deeper. So these were very on the surface, but just to show you microbial fluctuations in different processes. Now I want to go just a tiny bit more into the data of wine production. So it's a really fascinating process to make wine on many different levels, but also on the microbial level, actually. So in the end, you need first the yeast to grow and to metabolize so that they make the alcohol. It's the alcoholic fermentation. And then after that, it should be followed by microbes. So for example, this Œnococcus öni, so the wine is already in there. This does something that's called malolactic fermentation, and this is for the taste of the wine, really important. And so you need to have a kind of complex ecosystem in this production process. You first need the yeast, and then you need the microbes, and you need the right kind of both of them. And we used online flow cytometry to follow this up. So here we look at the bacteria, the Œnococci, and we use the staining for total and intake that I showed before. So these cells were, you get these packages from manufacturers, and then you put them in your process, and they get rehydrated. And we wanted to know, how does this change over time and once they start to rehydrate. And we were able to quantify the total and then the damaged population. What you also see here, that's typical of flow cytometry, often you have background noise that you need to make sure you tell apart from your cells. And then what we did here is to follow the growth. Here we're looking at the yeast actually. And you see the total and the intact population, and you see a lag phase, and then the cells start to grow, and then they slow down a little bit. So we were able to follow this in real time. Okay. Now let's go a bit more in that type of biotechnology that you see here. So this is really bioreactors. And this is a very simple example, but I think it's kind of beautiful data, so I wanted to show it. This is simply 2 E. coli strains. No, it's all E. coli strains, but they are grown in different growth conditions. So you see the blue data, it shows a really rich media. And then the red one, it was a less rich media, and you see just how the growth rate really declines, but in the end they reach the same cell density. In the gray you see kind of this thing that's called dilution, and that's more about the mechanics of it. It's not so important for the data, but I do want to show it because looking at this curve, it looks quite simple, but when you look at the y-axis, you realize that we cross here several orders of magnitude because this is a log scale. And that's microbes. They grow from low numbers to high numbers exponentially very fast. And this is not so easy for sensors to cover that full range. So often you only have a limited linear range that you can work with. It's similar with the flow cytometer, that there's a limited range where concentration can be quantified reliably. But what we do with the online is that we automatically dilute and we make sure we always stay in the linear range, and we do that seamlessly, as you can see here. So this is really, you can imagine you have a bioprocess, you want to understand how fast your microbes grow, and maybe you want to look at a design of experiments where you play with different types of media or different concentrations of nutrients, and you want to understand what effect this has on your process. So this is where this method can be super helpful. Now I want to go to a method that we published 2 years ago, is it now? It was also funded by this EU project, Promicon, that I mentioned. And here now we go really into something that's called fingerprinting. This is also something nice a flow cytometer can be used for. And the starting point here is a community of microbes. So it's not that we just have one strain, but here we had 3 different strains, and this is also called a consortium. And in microbes, that can be used to do very complicated processes that maybe one microbe by itself might not be able to do. This is very similar to the wine that we talked about, right? You need the yeast and you need the bacteria to get to the final product. And the fingerprinting in this case is to differentiate these strains and get the ratio of each of the strains during the process. So we had the community, then we had one of our devices for automatic sampling, automated fixation. So this was a kind of a more challenging protocol than the CyberGreen I mentioned before. It also has fixation steps, but we could do these automatically. And then it was measured in the flow cytometer. There was automatic gating, and cell counting and the fingerprinting of the populations. And yeah, so I will show some plots here, but not go in all detail. Please read the paper if you're more interested. So these are, again, these typical flow cytometry plots. Here you see a thing that's called scatter. So we're not just looking at fluorescence, we're also looking at scatter and some type of fluorescence. This is DAPI, which is linked to the DNA content. And then you see a lot of populations popping up. And these populations are, A and E, this is the same strain, but one's measured online and one's measured, manually prepared. And in the end, for as you go along, you see the different strains that we measured, and we were able to automate this protocol in a way that the online and the manual really gave the same results. And then, oh yeah, I forgot to mention this. This is with the group of Susanne Müller from Helmholtz Zentrum in Leipzig in Germany. So she's a really big researcher on flow cytometry. And we also have a new publication out with her, and it just was posted on bioRxiv last week, I think. And this one is about tracking the replication, the DNA replication of the cells. So imagine you have your cell culture, and you want to understand if all cells are equally active, or if there are some that are, in a resting state, because this can have effects on your efficiency or productivity. And for environmental microbiologists, this is also really interesting to understand who in a community really is active and is able to grow. And for this, we used a double staining method. One is, again, DAPI staining. This one will stain the DNA and will be used to quantify the total cells. And the other one was quite a complicated staining. It uses a fluorescent marker called Alexa 488, and it's able to bind specifically to newly synthesized DNA. So for this, there was a DNA analog called EDU fed into the medium for a certain amount of time. And so the cells that are active at that moment, they will incorporate the EDU. And then this Alexa 488 can bind to the EDU using click chemistry. So this is a quite advanced staining that we implemented here. And then from that, you can calculate the DNA replication percentage by dividing the number of Alexa 488 positive events, so the cells that have the EDU, the DNA analog, dividing that by the number of total cells, so the DAPI positive cells. And we did this for a couple of experiments. Yeah, I cannot show all detail, but, and you see it's complicated graphs because we measured a lot of parameters. So in the end, what you see is different dynamics again. So here we have one strain, and we can see the OD and the cell growth slowly increasing. And then we have in green, the DNA replication percentage. And then what's interesting, when you compare different strains, you see that they have kind of different strategies of growing. So the E. coli here in the middle, it seems that first they really replicate the DNA, and then they start to divide. So you have a peak in the replication, and then after that you have a peak in the cell numbers, also the strain down here. So I think it's quite interesting data, and feel free to check it out. Okay, I'm almost at the end. So I'm finished with all of my microbe examples. And I just want to mention that at the moment we are working on a project on using CHO cells. So these are cells, mammalian cells, that are kind of the workhorses to produce monoclonal antibodies. And we have a project ongoing with a Swiss research group here. And this is just a glance, so again, you see a flow cytometry plot. Here we have a staining assay where we can look at apoptosis. So this is one type of path that the CHO cells can take to die, let's say. So it can be quite relevant to keep an eye on the apoptosis, to make sure that the apoptotic population doesn't grow too high. Apoptosis can also be reverted if you're made aware of it. So it can be relevant for bioprocesses to monitor this and to react in real time. It can be a KPI, for example. And this is now the first protocol that we implement online, but there are many more that could be implemented and used. Okay, I talked a lot, and I showed you a lot of examples and a lot of data. And towards the end, I just want to take one step back to highlight that what we are talking about here is a measurement method that really produces big data on biology. it's maybe not as much as sequencing, but it is a lot. And this is because, as this example on the left shows, within one measurement, you already get a lot of information on each cell. So yeah, typically we collect at least 100, but up to 100,000 data points within one measurement. And each data point has several dimensions. So you can look at different fluorescent channels. You can look at height and width and area of the signal. So you get, 10 dimensions, let's say, on each cell. And at the moment, the way we do it and the way that many people do it, also in the examples I showed, we're really just using the tip of the iceberg, right? With this gating, we kick out and we disregard a lot of the complexity that's actually in the data. And I think a lot more of that could be used with AI and so on. And then if you run this whole measurement method online, you collect a data point, let's say every 15 minutes, you multiply the data. So you really get a really deep insight into your process, potentially. And this is something that it's just food for thought. I think this is really interesting and maybe something where flow cytometry can be used actually a lot more for understanding processes in a way that we might not really take advantage of at this moment in time. Yeah, so I want to conclude. I hope I could convince you that flow cytometry is a great tool for rapid measurements of KPIs and CQAs. And yeah, when it's complemented with this automated sampling, it can be turned into a PET technology. It's robust technology. It's very well established, but it's really versatile. So I showed you many different examples. So can be used in many different types of processes, and it can be used for many different types of parameters. So if you're not working with a flow cytometer yet, I hope you you consider it. And if you come across one, I hope you have my talk in mind and get inspired and have ideas on what to do with it. Okay, I'm done. I think I'm good in the time, and I'm happy to take your questions.
All right. Thank you very much for that very informative presentation. We'll now start with the Q&A portion. So if you do have any questions, please put those into the Q&A box and click submit. So we'll go ahead and get started. So the first question is, how does online flow cytometry compare to other monitoring methods like Raman or impedance-based sensors?
Yeah, so it goes a bit back to the overview slide I showed at the beginning. So I think Raman and impedance-based sensors, they can be used for some of the parameters that a flow cytometer can read out as well, like total viable cell concentration. I mean, Raman is also, you can measure a lot of biomolecules with it, but they are a bit more, they are less direct, I would say, than a flow cytometer. The benefit that they have in some way is that you don't need a label. So it's really nice also to have label-free technologies. It makes everything less complicated when you don't need to use the chemistry. But I think they are more limited maybe in the range of really biological characteristics they can measure. So always, every method has its pros and cons.
All right, thank you. How do you deal with cells of different sizes in a specific sample?
So there's we will see that they have different sizes. So you can see that either by looking at the scatter or also by staining, and then like the example I showed with the large and the small bacteria. So in these examples, often we are able to really differentiate, and we would be able to tell with a flow cytometer what's the big population, like how big is the big, what's the ratio of big and small. So in principle, they can all be measured and differentiated. of course, there are always size limits. So there's a maximum size of cell that will no longer fit into a flow cytometer, let's say, and that depends a bit on the setup. I'd say 50 to 100 micrometers is probably the upper limit. And then there are also lower limits where it's really hard to measure super small particles. But we've measured, I think, down to 0.5 micrometers.
All right. Let's see here. The next one we've got is, where do you see the biggest use case for online flow cytometry and monoclonal antibody production?
Yeah, that's to be found out. So there are many types of staining assays in principle available. So, and then what's the biggest use case? It will depend on the biggest need from people who run these bioprocesses. So I think one hot topic right now is perfusion. So where you really run a more continuous bioprocess. And there, for example, shear stress might be interesting. So that's something you can quantify with a flow cytometer, and it will help you to kind of make sure that your stirring is good enough to get oxygen in, but not so high that you would stress your cells. So this is something that could be used to monitor the process. Others might be cell cycle, or apoptosis.
All right. Can you monitor any cell characteristics, and what are the limitations?
So you can monitor anything that you can stain with a fluorescent stain. And so as long as there's no stain around, you might not be able. And of course, it's easier when there's already commercially available stains, and there are for many, many things. But I'm sure there are a lot of, chemical and biological limitations. So very rare events might make it hard, or very subtle differences might also make it hard. But overall, I think there's a big bucket of things out there that can be looked into.
Got time for one more. Go with, can you quantify smaller biological particles like a virus?
Yeah, virus indeed, it's too small. So it's a bit like I said before, that there is a lower size range, and it's just the physics of the optics. You come in a size range where the laser has kind of the same size. The laser light has the same size as the particle, and then it just cannot be quantified anymore. So unfortunately, a virus cannot be measured with a flow cytometer.
All right. Well, thank you so much for your time today and your research. Before we go, I'd like to thank the audience for joining us and their questions. Any questions that we didn't have time for, or those that are submitted during the on-demand period, will be addressed by the speaker via the contact information you provided when you registered. This webcast can be viewed on demand for a year until April 2nd of 2026. LabRoots will alert you via email when it's available for a replay, and we do encourage you to share that email with your colleagues who might have missed today's event. So until next time, thank you once again, and goodbye.